Imagine a groundbreaking discovery that could change our understanding of brain development forever. Recent research has unveiled that immune cells in the human brain play a pivotal role in shaping its growth during the prenatal period. This revelation suggests that these immune cells are not just defenders against disease, but also crucial orchestrators of brain cell formation. Inhibitory interneurons, a specific type of nerve cell, are estimated to constitute between 25% and 50% of the neurons in the adult cortex, the wrinkled outer layer of the brain. Remarkably, humans possess more than twice the number of these interneurons compared to mice. These cells are essential for relaying signals between other neurons and maintaining balance in brain activity through a chemical messenger known as GABA. GABA acts as the brain's primary inhibitory signal, reducing neuronal firing and counteracting the excitatory signals that can lead to overstimulation. Disruptions in the function of interneurons have been linked to various neurological disorders, including epilepsy, autism, and schizophrenia. A study published on August 6 in the journal Nature has shed light on the mechanisms that drive the proliferation of these interneurons in the developing human brain, suggesting that this process may be unique to our species. This is why traditional animal models fall short, explained Daenkun Yu, a co-author of the study and an assistant researcher in pediatrics at the University of California, San Francisco, UCSF. To investigate this phenomenon, the researchers created an organoid, a miniature, three-dimensional structure derived from stem cells that simulates a full-sized human brain structure. Prior to this organoid study, research involving laboratory animals indicated a connection between the activation of the maternal immune system during pregnancy and a reduced number of interneurons in the offspring's cortices. Such immune activation could occur due to viral or bacterial infections. In earlier studies with lab mice, the researchers identified microglia, the brain's resident immune cells, as a key factor in this relationship. In recent years, scientists have increasingly recognized the parallel development of the immune and nervous systems. Dr. Shanhua Piao, another co-author and a physician-scientist specializing in neonatology and developmental neuroscience at UCSF, noted, "Microglia play a crucial role in fine-tuning and regulating nervous system development. This study adds a new layer of understanding to their functions." Building on previous research, the team developed organoids that mimic a critical structure in the fetal brain, from which many cortical interneurons originate. This structure occurs around the 8th week of pregnancy and disappears approximately 8 months after birth. Notably, the researchers successfully incorporated microglia into their organoid model, a feat not previously accomplished. The findings revealed that microglia in these organoids were a significant source of insulin-like growth factor 1, IGF-1, which is instrumental in driving the rapid increase of interneurons during early brain development. When the researchers inhibited IGF-1 signaling, they observed a halt in the proliferation of interneurons. Interestingly, when they deleted the gene responsible for IGF-1 production in microglia within a mouse model, there was no observable change, suggesting that this process may be uniquely human. The researchers concluded that these findings indicate an evolutionary adaptation of microglial function to meet the heightened demand for interneurons in the human cortex. This insight could provide a glimpse into the evolutionary traits that contribute to our distinct cognitive abilities. However, it is essential to note that organoids are not perfect replicas of the human brain, and their capabilities are limited. Currently, these models are effective for studying the early stages of development, but they do not accurately represent later stages or the complex circuit-level activities of the brain, Yu explained. Future research may further illuminate the previously unrecognized role of immune cells in brain development. Nicoletta Linnaeus, the Health Channel editor at Live Science, has a background in neuroscience and science communication. Her work has been featured in various reputable outlets, and she remains actively engaged in the dance community in New York City.